second block of 2 talks. And my name is Henk Stoff, for the people that don't know me, but of course this is Ike's day, but I was a little bit involved also with the program, and so he asked me to introduce these 2 talks. So Rutger Hermans is the next speaker, so he is talking about a biological subject, but I understand he came from physics, and maybe he's aware of a very famous book in physics by Nico van Kalken. It's a famous theoretical physicist here from Utrecht, and he has a very similar name, so maybe the title of his famous book maybe inspired you to this title, so stochastic process in biology, so please I give you the floor.
Yeah, Van Kampen actually is also in biology. I mean, many people in biology who work on stochastic processes study that book in a lot of detail. It's a bit frustrating because I really like the text of the book because he's very like lucid in his reasoning, not too mathematical and not too loose. But he hides, for instance, a lot of the important results in exercises without answers. So then you're in chapter 5, he's like, as we saw in chapter 2 in exercise 3, that nobody makes. I'll mention his name once in this talk, I think. What you probably also don't know is that I got my introduction to statistical physics from you in 2002, I think.
Okay, so
so if I make any mistakes, you know how...
it's my fault.
You know who to blame.
Now you know why I asked Henk to introduce you.
Okay. Yeah, there's not going to be lots. Anyway, when I was thinking about what to present today, I realized that there was going to be a very broad audience with people from many different fields. So I thought I'm not going to talk about a very specific topic of my own research, but instead give you a kind of a kaleidoscopic view with a couple of examples of how stochastic processes theory, so the mathematics of stochasticity, is nowadays used in biology. It has been used. And some of the examples will be sort of modern and others will be very classical. I apologize to people who know all about these things. And so basically the question that I would like to ask here is, why does biology need stochastic models in the first place? So the whole goal of this little talk will be to answer that question in 3 possible answers, like 3 different reasons. And I will illustrate each of these reasons with a different example. So the first reason, I know the text is a little small, it says many biological systems are plagued by noise. And they've often evolved to cope with this noise, and therefore we may need stochastic models to actually understand why they are designed the way they are designed, because they've actually evolved to cope with a noisy environment and with their own noisy behavior. And I'd like to give you an example by first looking at bacteria. This is actually something that I do know something about. Later I will bluff a little. No. Bacteria actually are very noisy, in a sense that if you use the word noise, I mean fluctuations, right? So if you look at genetically identical bacteria under a microscope, you'll see that their protein concentrations actually fluctuate a lot. So this is an example from a study from almost 20 years ago already, where these are E. coli cells from E. coli, and they're genetically identical for the most part. And what they build into them is 2 different colors of fluorescent proteins. So they produce proteins of 2 different colors. These are fake colors. In reality, they're yellow and green, but they filter them and they show them as red and green. So if these cells have a lot of both of these proteins, they become yellow, bright yellow. They have neither of them, they become very dark. And if they have one of the 2, they're either red or they're creamy. So you see that they have very different concentrations of these 2 different proteins. And also nowadays you can actually follow single cells under the microscope with a computer. You can analyze them to follow them over time as they grow and divide and grow and divide. And you can follow the expression, when I use the word expression, I mean the concentration of protein over time. You see that they actually fluctuate quite a bit, easily by like 50%, 30% maybe. So they fluctuate a lot. Now, why is this? Well, let's first. If you observe this, and people knew this for a long time, immediately a lot of questions come up. Like, what are the main sources of noise in gene expression? Well, why is this so noisy? What determines the distribution of these protein copying numbers? Which are the variables that play a role there? What's the effect of this noise on the functioning of the cell? If all your components fluctuate by 30% up and down at random, how can you still function? And what can a cell do to reduce noise or its susceptibility to noise, or to cope with it, to live despite that noise? And so over the past, I would say 20 years, a lot of people have done experiments, both experiments and a lot of modeling to answer some of those questions. And I can't review everything, I just want to give you a flavor. So just for the people who are not in biology at all, some refresher from your high school biology. So in the cell we have DNA, and genes are all the DNA, and by a process called transcription, RNA molecules are made from the DNA, from these genes, and these RNA molecules are then translated, a process called translation, into protein. And now it's important to realize that bacteria are really small. They're usually like 1 micrometer in their width. E. coli is about 2 or 3 long, so they're really kind of small. And hence the copy numbers of many of these proteins in one cell are actually very low. So for instance, DNA molecules, so each gene on the DNA is present in 1 to 4, in extreme cases 8 copies in the cell, so it's a very low copy number. And RNA, typical numbers, if you at any point of time look into the cell, somewhere between 0 and 20 copies of RNA per gene that is expressed. And then for the proteins, we have very different, very variable numbers. Some types of proteins are present in like 10 to 100 copies per cell. Some of them up to like 10,000. So it varies a lot. Some of them have very low copy numbers, and some of them have high copy numbers. And at the same time, they're also produced, because they're in such small numbers, they're also produced at very irregular intervals, or at a very low rate. So DNA, for instance, is typically duplicated, roughly in the time it takes to duplicate a cell, order of an hour, right? RNA, typically transcription rates are 0 to 5 per minute, right? So typically 1 per minute, something like that. And then every time you produce an RNA is kind of short-lived, so you produce it, and a couple of minutes later it's gone, and in that time, multiple rounds of translation have typically occurred, so you produce a burst of proteins. So typically, like if you make mRNA 0 to 5 times per minute, you get 0 to 5 bursts per minute. So together, that means that inside a bacterial cell, you're not in the large numbers limit, where rates are fast and numbers are large, and you can just forget about noise. That is totally the opposite. You get discrete events and small numbers that fluctuate quite a bit. So keeping that in mind, one way to model this particular source of noise, which was done in many different ways, so I'll take an example, 2006, is to say, well, if I know that this degradation of RNA is actually kind of fast, so I have rapid degradation, and in that very short time that an RNA lives, I produce a burst of protein, then I can model this like this. I have a rate at which I produce mRNA, rate K1, I model it as a Poisson process. And then at that rate you have a burst of protein production, and that protein is then diluted. Protein is usually in E. coli pretty stable, so it's mostly diluted by the growth of the cell. So if I then write that as a master equation, as Nico van Kampen would have liked me to do, is you'd have an equation for the probability of finding a certain concentration of this protein, and that concentration is the probability actually is changing in time, because of two processes, I have dilution at the rate gamma 2, which is in this term, which is modeled as a deterministic process. I'm just diluting at a constant rate. But then there's also jumps. So I can jump from a concentration x prime to a concentration x with a rate k1 times the probability of the size of the jump, which is the w function here. And that I can do for any jump size. So this is an equation that describes the evolution of the probability distribution. Now, the only thing you then need to put in is the probability of a certain jump size. And that you can actually measure. So people did that. You look at a particular cell and you count the number of, in this case, beta-galactoside molecules of one particular enzyme that has been labeled. Then what they could do is they could look in time. You see that for a long time it's constant. And then you get this burst that I talked about. And it's constant for a while. There's a burst, constant. So from this type of data, you can actually deduce the distribution of these jump sizes, these burst sizes, and it's roughly exponential. So now we have a full model because we have this set of equation. The only thing we have to put in is this function. We actually have it. And then the prediction, in this case, well, let me go back. So what you're interested in, if you look at a population of cells, is the steady state distribution P of x. So it means that you're interested in on the long run, this probability distribution will become stable. You set this equation to 0 and you solve it. And it turns out that in this simple case, it is a gamma distribution. The steady state distribution is a gamma distribution. That's something that people know about, and you know what it looks like. So now, a couple of years later, people actually measured these distributions for many different proteins in the cell by labeling them one by one with a fluorescent label. And indeed, many of these distributions, not all of them, certainly not all of them, but most of them, fit the gamma distributions very well. So that gives you an idea that at least this type of model can describe, doesn't prove that it's the correct model, but can describe these type of distributions. And the nice thing now is that these parameters that you get out, the gamma distribution only has two parameters. One of them is essentially the burst size, the mean burst size. And the other one is essentially the rate at which you produce RNAs. So they are actually meaningful parameters that you can then study and verify by other means. Okay, now so the cell, only by the fact that it has a random production of mRNAs essentially, and a random burst size, that already explains basically these pretty wide distributions. That's the point that these models make. So the cell somehow has to deal with this. So it could come up with ways to reduce this noise, or it could come up with ways to filter noise that it doesn't want later on in the process. And well, many papers have been written out ways the cells can actually filter out noise or deal with noise. I just want to give you two patterns in the network that are in the sort of gene regulatory network that occur very frequently, and that can be shown to reduce noise. So one of them is kind of obvious. It's called negative feedback. So if I have some signal that is fluctuating, and it's itself produced at some rate, but now I impose an interaction such that if this signal fluctuates up, it actually reduces its own production. So this is what the arrow here means. It actually represses its own production. Then you can easily understand that it will filter out certain frequencies of noise. If this signal goes up, it starts to suppress its own production, and therefore it doesn't go up any further. It's reduced further down. And that you can actually test in experiments. For instance, you can take a gene that you build it into an E. coli cell, and you look at the distribution of its expression. And now you engineer that this gene actually binds to its own what we call promoter, the place that decides how frequently it's being transcribed, and by that way it represses itself. And then you can see that indeed there's a much narrower distribution in the data. Now, there's all kinds of other ways you can filter out noise. So this is a more fancy, I think, way of doing it. So suppose I have a fluctuating signal S, and it positively affects another signal A, some other molecule, some other concentration A, but also B. And now I'm going to use B as my filtered signal. So this is going to be used in the cell to do something. And I want this to be a kind of filtered version of that one. So one way you could do this is by having this signal A and B, which now correlate with the noise in this first signal, bind to each other and produce something else, C. Or they might actually destroy each other. What happens now is that if this thing fluctuates up, both A and B go up, and they tend to bind more frequently to each other to produce C, and as a result, B is kind of sort of has a sort of quenched version of the fluctuation of this kind. Because if this one fluctuates up, both of these go up, and B is degraded more rapidly. But this actually also works, and it has been suggested at least that it's working in some mammalian systems, some animal systems, where microRNAs might do exactly this. So microRNAs are small pieces of RNA that are being produced not to produce protein, but to do something other than that, usually regulation of other proteins. And one thing that it could do, for instance, is some transcription factor, some regulatory element in the cell fluctuates, but instead of only regulating the gene that it's supposed to regulate, that it's supposed to affect, it also affects a microRNA, which then binds the mRNA of this target gene and degrades it. So what happens now is that if this thing fluctuates up, both the target gene is at risk of going up, and the microRNA. Because both go up at the same time, this one degrades part of those and basically takes out part of the fluctuations. This also you can do, the models have been written down, full stochastic models to characterize these systems to see which fluctuations they would take out, which, what kind of bandwidths they would take out, and which they wouldn't. So, okay, so that was the first example, and I'm almost through. No, I still have some time. So, all I wanted to show with this example is that actually biological systems are very noisy. I could have taken many other examples. And that often this noise is actually detrimental. You don't want it. And therefore the cell has actually built in sort of mechanism to deal with it. Yes.
Sorry, but these processes that you mentioned at the end, are they also described with the master equation? Or is it a different story?
You can, so you need different models for it, but yes.
So just that you change the kernel in the integral on the right side.
Oh, so you need to put in more. So that master equation that I gave you was just pretending that one gene stands on its own, has nothing to do with all the other guys. But of course it becomes a lot more complicated if you have many different genes that actually affect each other still switching. But you can still, in principle, do it. Solving them gets a lot harder. Okay, now I want to jump to an entirely different reason why you want stochastic models in biology. And this is that noise can actually qualitatively change the dynamics of a system. So if you don't put it in, you might get the wrong conclusions. So that's a very different reason why you need stochastic. And so I'm jumping to an entirely different subject here, which is a very classical subject, I would say, which is predator-prey cycles in biology. So this is a very old subject in biology that people in the 1920s already studied. So this is a really famous example of a lynx actually chasing rabbits. What people found out in the 19th century is that if you looked at the population of those lynx and those rabbits, that they showed these huge fluctuations over time with a sort of regular interval. And typically, the predator goes up first, and the rabbit, so in this case the prey goes up. That allows, that's at least a naive interpretation, the predator to actually increase its population size because it has food, but that then leads to a destruction of this population, and it goes on like that. And traditionally these models have been described, or these systems have been described by deterministic equations, like differential equations that produce cycles. But it turns out that it's actually not so easy to get realistic models that have sustained cycles. Often people present the Volterra models. These are really old and well-known model, but it actually turns out that that model is kind of a singular system. If you slightly modify it, what you get is not a sustained oscillation, but a spiral to a fixed point. So here's an example. If you have rabbits and lynxes that interact with each other, so the rabbits just reproduce logistically, which means that they grow exponentially until they reach a carrying capacity, and then their growth rate goes to 0. And they can be caught by these lynxes, L, at a certain rate. And the lynxes actually grow if they catch rabbits, but they also die sometimes. If you do this, you don't get sustained cycles. What you get is a spiral, so the lynxes and the rabbits, they oscillate for a while and get to a fixed point. And it kind of makes sense because there's not really a reason why it would overshoot all the time and keep on going. Right? Now, lo and behold, if you take this model, this exact model, but now you write it in terms of a master equation, so each of these processes here now becomes again a Poisson process, stochastic process, and you simply run a Gillespie algorithm, as they call it, just run it in a simulation, you get oscillations, and large ones. So here, this is what these authors produced. You get irregular, but still clearly a certain frequency out. So how did they do it? So they run these simulations in as a so-called Gillespie, or I think people also call it kinetic Monte Carlo type algorithm, to exactly simulate a trace from a master equation. And you find these oscillations with amplitudes that are much larger than you would expect based on just the Poisson noise in the system. So why is this? Well, you can analyze the traces of your simulation, or in fact you can calculate from the master equation the power spectrum of the signal, which is simply how frequently or how strong does a certain frequency occur in your signal. And you can really see that there is this huge peak over here, which we call resonance. So even though nobody puts this frequency in the system by hand, you're not driving the system at this particular frequency, somehow the system picks up by just the randomness of birth and death processes this particular signal and amplifies it hugely. So it's called a resonance. And that of course has to do with the fact that you have these decaying cycles over here, right? So you drive the system with random frequencies, you make it wiggle, because it has this intrinsic periodicity inside, it actually resonates and produces these oscillations. So how do you do this? Just to mention Nico van Kampen, what you do is you take the master equation, and then you do the omega expansion, van Kampen omega expansion, you end up with a Langevin equation, and from there you can actually do the creation. So now at least I've mentioned van Kampen. So this was one example, yeah.
It could also be that there are time lags in the population structure, right? With young animals, all animals, they don't reproduce immediately.
Yeah.
Don't
So
catch prey, etc.
in this model, we didn't put any time lags in, but obviously this is not the only explanation that you could give for oscillations, and indeed, like the delay equations could give you oscillations. I think there are models where the lynxes at some point have enough food, they just stop eating, and that also helps. So there's all kinds of ways to get it. But the only thing that I'm trying to say here is that some observations that you see in nature, or basically in any stochastic system, may not be there deterministically, may not be there in the limit of large n, basically, but be only there because of resonance frequency and the intrinsic noise. And that is a qualitative change that you would not have been able to see if you only did deterministic modeling. All right, so now I'm at the last part. I probably have to be a little quick there. There's a third reason why you need stochastic models, I think, and I like that one. And that's because stochastic behavior can actually emerge by natural selection. So natural selection can actually select random behavior in organisms. And there's actually two ways that I can think of that this can happen. Maybe one is a subclass of the other, but one example of this is at least suggested to be this. It's called bacterial persistence. So what is this? If you take E. coli and you subject it to antibiotics, for instance ampicillin, then most of the bacteria will actually die. But somehow there's a certain group that has been known for a long time, had been known for a long time when the paper came out. There's always a certain number of cells that somehow survive. And they were called persistent cells. And what these authors in 2004 showed is that if you actually look at those cells, so what they did is they made a device where you can grow cells in sort of grooves so that they stay in line. So they grow and they push each other out along these lines. And what you can now do is first observe them in just growth medium without antibiotic, and you see how they grow. You can actually see which cells grow and how fast they grow. Then you introduce ampicillin, you see them die, because they lyse, meaning that they pop open and they're gone. And then you can ask, is there something special about those cells that don't die? And it turns out that the cells that don't die are the cells that in earlier times, right before it, already didn't grow. So somehow in this population, there's a certain fraction of bacteria that just decide to stop growing. And those are the cells that later on, when you apply the antibiotic, stay alive. Now, if you take the ampicillin away, you might think these cells are sick or dead. No, you just wait a while, suddenly they switch and start growing again at the same rate as the other guys did before. So there seems to be a stochastic process going on, and they measure the rates at which this thing switches between two states. Maybe more than two states, but at least two states, one growing state and one non-growing state. And in this case, it seems that it's actually useful to have this really small subpopulation of cells that doesn't grow. They offer, how to say this, they sacrifice part of their fitness, but there is a certain probability at some time that you meet some agent that kills you if you're growing. And it's useful to have a subpopulation that survives at that point.
But these cells may be resistant to the antibiotic.
Yeah, so they're not.
To compete very well in the natural population.
Yeah, so the point is that if you take these cells here and you just let them grow out and you repeat this thing, you get the same thing. The ones that are growing rapidly will die, and the ones that are not growing, they will survive. So you can repeat the experiment with those cells and nothing has changed. So there is no genetic difference between them.
Okay.
So people have then asked this question, like, is it true that under some circumstances it is useful to have some subpopulation in one state and another subpopulation in another state and randomly switch in between to anticipate possible switches in environment that require you to be in the other state? And this is actually a kind of complicated model, so I won't do all the derivations, and there's no derivation at all. But the idea is basically that you have some environmental signal that fluctuates between different types of environments: the blue environment, the green environment, the purple environment, etc. Generally, there's a certain number of environments that we think of as having a color. And this is a random process. And then in these different environments, there are different phenotypes that are optimal. So you can think of this as a blue genotype, a green phenotype, a purple phenotype. So now the question indeed is, what is optimal to do? Should a cell decide just on one phenotype, maybe the black one, and just stick there, and just accept that if it's in a different environment, it doesn't grow very well, or it's dead, in fact? Or is it better to do a bet hedging strategy, so to hedge your bets and to say, well, I randomly switch between those phenotypes so that in the population there will always be types of that are okay in every environment, but typically are not in the right phenotype. So what is better? And you can model this, you can do the calculations, and it turns out that the optimal behavior in this case is a clever bet hedging strategy, where you actually switch between those phenotypes such that you are, well, essentially, if on average the environment is often in the green environment, you should switch to that environment more frequently and leave it less frequently, basically. So the statistics of your own switching should be matched to the frequency of the environment. But if you do that, then it's actually better to have the bed hedging strategy. And it works like this. So if I initially am blue and I'm very happy in this environment, the population grows out. And in the meantime, some of these guys switch to the wrong phenotype. And that's a shame, that costs you fitness. Some of my children will be lost because they switched to the wrong phenotype. However, when we suddenly switched to this green environment, and I couldn't see that coming, now at least one of my offspring in this environment, at least some of my offspring will be in the right phenotype, and they will grow out and take over the rest of the population. When the next environment comes, I will be prepared by at least some of my offspring. And you can do what they actually do is they calculate a Lyapunov exponent to see which strategy gives you the highest long-term exponential growth, essentially. And this turns out to be the highest growth. You can actually also compare it to sensing your environment. So what if I can actually measure in which environment I'm in and then switch to the correct phenotype? That would of course be better. Unless having the machinery, like the sensory system, to do this costs me something over the long run. I have to express a certain, if we think of bacteria again, I have to express a certain sensing system, certain enzymes, and that costs resources. And now, what will be optimal now depends on all kinds of parameters. So the cost of the machinery involved. If that cost is high, then certainly it's better to be a bed hedger, just to forget about measuring and do the random thing. If the speed of the response is actually low, so even switching takes you time, even if you measure and then switch. If that is a slow response, of course, that favors bed hedging. If the frequency of the environment's switching is very high, well, then probably, so if it's low, it's nice to be a bed hedger because you don't pay the cost of having the machinery, and for long periods of time, most of your population is in the right phase. And actually, if you do the calculation as they do in the paper, it turns out that there's another thing that is very important, and that's the uncertainty in the environment that you can actually, it comes out of the model as being a kind of entropy. So it basically says that there's an environmental entropy, which is how uncertain am I about the environment that I'm in, which is how frequently am I in a certain environment, but also how unpredictable is the switching between environments. Like if environment J always follows I, then that helps the bed hedger a lot, because it can also predict by random switches where it will go to without sensing. If this uncertainty is really high, then you need a sensing machinery to do it. Right. So now I think my time is basically up, right? Mm-hmm. Yeah. Then I will only say that in some cases in evolution there is no you can't really talk about the optimal strategy being chosen, because it's more like game theory. But the question is which strategy that you actually use is stable against other invaders. And that doesn't have to be anything in terms of optimality for a population or for the individual or something like that. So it's a bit like the measles outbreak now in the US. Like there is no stable way. Like if everybody is
vaccinated.
Sorry? Yeah, if everybody is vaccinated, then there is actually an incentive not to be vaccinated, because there's a risk involved, a small one. There's a risk involved in vaccination. But if nobody vaccinates, you have a lot of measles outbreak. There's a big incentive to get vaccinated. So that means that there's no stable single strategy that would work in society. So the same works in evolution. So sometimes the best thing to do for a population, even for the individual, is all to be vaccinated, so to say. But that is not a stable thing to do. And what would be stable then is if everybody would be vaccinated with this probability P randomly, and this P is chosen exactly such that the risk, the payoff of getting vaccinated or not getting vaccinated is exactly the same. So the risk of getting the measles is high enough such that it pays off compared to the risk of getting the vaccine. In that case, nobody can invade the population, and you get what they call is a mixed strategy, random strategy. And there are many examples in evolutionary biology where the only stable thing to do is to basically be unpredictable, so that others cannot come up with a strategy to beat your predictable strategy. So that's another way that stochastic behavior can actually emerge as a mixed evolutionary stable strategy. Okay, so that actually brings me to the conclusion slide, which is the same as the slide as you've seen all along. So, why do we need stochastic models? Because systems are noisy and have been evolved to deal with it. Because noise in models can actually change the behavior of a system qualitatively, and because there's actually functionality in certain stochastic behavior in biology because it can be selected for by evolution. So thanks.